Welcome to Bosker Sciences. Today's topic is going to be on biological macromolecules. So the four things that are going to be covered in this video is the structure and function of carbohydrates, lipids, proteins, and nucleic acids. Three things that you should know before you watch this video are the difference between a monomer and polymer, dehydration synthesis, and hydrolysis. The difference between a monomer and polymer is that a monomer is a type of molecule that has the ability to chemically bond with other molecules in a long chain, while a polymer is just a chain of monomers. Dehydration synthesis is a reaction used to build polymers from monomers, while hydrolysis is a reaction used to get monomers from polymers, and I described it more in detail in my previous video. The first biomolecule that we're going to talk about is carbohydrates, and it consists of carbon, hydrogen, and oxygen. The ratio of hydrogen atoms to oxygen atoms is always 2 to 1, and there are three classes of carbohydrates: monosaccharides, disaccharides, and polysaccharides. Monosaccharides have a chemical formula of C6H12O6, and an example of these include glucose, fructose, and galactose, which are isomers of each other. If you don't know what an isomer is, it is organic compounds that have the same molecular formula but have different structures. Disaccharides have the chemical formula C12H22O11 and are made up of two monosaccharides joined by dehydration synthesis. And if you notice, it should be C12H24O12, but a water was lost in the reaction. Next is polysaccharides are polymers of carbohydrates and are formed as many monosaccharides joined together by dehydration synthesis. Next is carbohydrate function. The body uses carbs for short-term energy storage and cell structure. The two types of fuel carbohydrates are cellulose and chitin. Chitin makes up the exoskeleton of arthropods and fungi cell walls, while cellulose makes up walls in plant cells. There are two types of fuel storage carbs, and they are starch and glycogen. Starch is plant energy storage, and a couple examples are amylose and amylopectin. Glycogen is animal energy storage, and it is stored in the liver and skeletal muscle. The next biomolecule is lipids, and what you should know about lipids is that they don't have a monomer, but they're mostly made up of fatty acids, and they include fats, oils, waxes, and steroids. They're all hydrophobic, and hydrophobic means water resisting. Most lipids consist of one glycerol and three fatty acid chains. And a glycerol is an alcohol, while a fatty acid is a hydrocarbon chain with a carboxylate at the end. There are two types of fatty acids, which are saturated and unsaturated. Saturated fatty acids only have single bonds between carbon atoms, so they are straight and remain solid at room temperature. Unsaturated fatty acids have double bonds and are liquid at room temperature because of those kinks, as you can see at the bottom. The functions of lipids include long-term energy storage and structural. Phospholipids are a major component of the cell membrane, and cholesterol is important for the fluidity of the cell membrane. Phospholipids are modified lipids which consist of two fatty acids attached to a glycerol backbone, has two hydrophobic tails and a hydrophilic head. And I would memorize the difference between hydrophobic and hydrophilic because it comes up a lot in biology. And another function of lipids include endocrine, because some steroids are hormones. The next biomolecule is proteins, and it is made of carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Polymers/polypeptides consisting of units called amino acids are joined by peptide bonds. Amino acids consist of a carboxyl group, an amine group, and an R group, all attached to a central carbon atom. The R groups are characterized by their chemical properties, and the interactions between these R groups determine the structure and function of the protein. And there are four levels of protein structure, which are primary, secondary, tertiary, and quaternary. Let's talk about the four levels of protein structure. The first is primary structure, and it is determined by the sequence order of amino acids. In the secondary structure, it can either be an alpha helices or beta-plated sheets. In tertiary structure, it is a 3D shape of the protein and minimized free energy, So there's a smaller loss of energy through like friction and heat, like stuff like that. And the bonds in tertiary structure include hydrogen bonds, disulfide bridges, ionic bonds, and hydrophobic interactions. The fourth is quaternary structures, and it is based on interactions from multiple polypeptide units. Proteins have multiple functions, and the main one is growth and repair, but is also involved in regulation, like insulin, which lowers blood sugar, and movement, which is like actin and myosin, which is responsible for muscle contraction, and it is also responsible for signaling from one cell to another. And proteins also make up enzymes, which catalyze chemical reactions. The last biomolecule is nucleic acids, and they are made up of carbon, hydrogen, oxygen, nitrogen, and phosphorus. It has a 5-carbon sugar, a phosphate group, and a nitrogenous base, which is like adenine, guanine, thymine, and cytosine, and uracil in RNA. The monomers of nucleic acids are called nucleotides, and there are 2 types of 5-carbon sugars, which is ribose and deoxyribose. The carbon atoms of deoxyribose are numbered 1 through 5, and DNA consists of 2 antiparallel strands running in the 3' to 5' direction. And you don't need to know about this yet, but it's important in DNA replication. Nucleic acids only have one main function, and that is the storage and expression of genetic information. For example, DNA encodes the information the cell needs to make proteins, and RNA converts the information in DNA and does the actions for making a protein. And if you need to see how RNA and DNA look, you could take a look at the picture on the right. So that wraps up today's video, and tomorrow's video is going to be on cells and their organelles.